Our lab studies neurodegenerative diseases, and in particular, Alzheimer's disease. Our specific work involves using postmortem human brains. These are folks who've been generous enough to donate their brains to science, and so we're able to use their brains to better understand how Alzheimer's disease happens and sometimes why it doesn't happen. One of the reasons Alzheimer's is an interesting disease to research is that it's so common. It affects millions and millions of Americans, and yet we don't have a full understanding of what causes it. So one of the things our lab is involved in is generating what we call a polygenic risk score. So this is using a number of these genes that we think confer risk for Alzheimer's disease, checking whether you have what we call the risk allele. That means you have more risk of having Alzheimer's disease. And then we find the people who have more risk and the people who have the least risk based on their genetics. And we're comparing them to try to understand the molecular biology and cellular biology underneath that response. Working with HiAC has helped us in a couple of big ways, I would say. One is that we know our data is really safe, which is really important when you're working with humans. It's very precious. Also, because we're using single-cell RNA sequencing, we are generating terabytes and terabytes of data from human individuals' brains. And since we have a single repository for our data that everyone in the lab can access at any given time, we can set our scripts to run and process multiple data sets at once. So just the efficiency with which we can analyze these very large data sets is massively improved. So since our goal is to develop therapeutics for Alzheimer's disease, the faster we can do that, the better we can contribute to the research that's going on in the world to better understand this disease and better help the patients that are suffering.